Greeting and thanks to the organizers and participants of this conference. I am Farideh Orouji, Master of Genetics from Shiraz University of Medical Science, Iran. I want to talk to you about evaluation of interleukin-22 gene polymorphism in 867,810,424 coming AG and 1,390,124,543 AG. Positions in women with infertility. Infertility is the absence of a couple for 1 year. The main disorders of infertility are pathological spermiogram, ovarian problems, fallopian tube disease, pelvic adhesions, endometriosis, cervical factor, or idiopathic cause. The number of follicles per person is genetically dependent, and infertility is unknown due to immunosuppression and decreased expression, inadequate function, or reduced access to lymphocytic and chemical agent in the uterus. Some immune cells of the reproductive system, such as T and K, dendritic macrophage cells, are involved in the endometrial secretory stage, decidual angiogenesis, trophoblast migration, and immune tolerance in pregnancy, and dysfunction of these cells with infertility, abortion, and other complications of pregnancy. Inflammation is a non-specific, acute, and chronic immune response that affects ovulation, hormone production, as well as endometriosis and infertility. And the placenta, as a member, has the ability to produce immune cells. Structure and function of interleukin-22 and its role in the incidence of diseases. The primary structure of interleukin-22 is similar to the anti-inflammatory cytokines of family 10 and as a pro-inflammatory cytokine. The main sources of this protein are a set of NK cells, hematopoietic stem cells in the bone marrow, myeloid cells such as macrophages and neutrophils. On the one hand, this cytokine stimulates inflammation of tissue and causes their regeneration. And on the other hand, it is involved in the formation of immune microenvironment outside the uterus by stimulating the production of cytokine 6 and 8 and vascular growth factors. Embryonic trophoblast exposed to interleukin-22 proliferate, and the expression of this cytokine is lower in spontaneous abortion specimen than in normal pregnancy. The important role of this cytokine is in the protection of the epithelial barrier and the care regulator of mucosal hemostasis, and in limiting inflammation and protecting the mucosa and its low levels. It is associated with several inflammatory diseases in the intestine and the excretion of connective tissue. This gene is located on chromosome 12, 12q15, and contains 6 axons and encodes a protein that acts on antimicrobial defense at mucosal surface and in tissue repair. The highest expression of this in this gene is in appendix, duct, duodenum, esophagus, testis, bone marrow, spleen, liver, and bladder, respectively. And in other tissue, the expression of this gene has not been observed. This gene has several single and multiple nucleotide variant 268 SNP, of which two variants are discussed in this article. Both positions are an SNV with alleles A and G, whose function is to splicing on introns predominantly and to create genetic diversity downstream of the gene. Length of this stand 29 pairs open. The interleukin 22 gene acts as autocrine and procrine and functions to survive, grow, differentiate, and activate tissue and cells. In this study, half a cc of the patient's venous blood was added to half a cc of EDTA, and then DNA was extracted from the peripheral blood by salts and depositions method, and then the concentration and quality of DNA were evaluated. The types of both polymorphisms were identified by PCR arms methods. Others were then amplified specifically and used to identify point mutation, and natural alleles were differentiated from mutants. Their results were taken on aqueous gel, and statistical analysis was performed. It is a method in which samples with an electric charge move under the influence of an electric field and separate from each other. Factors such as the nature of the sample molecules, electric charge and field current intensity, their size, environmental effects such as buffer type and heat during work are among the factors involved in the separation of sample molecules. Aqueous or polyacrylamide gel was used to separate the DNA molecule. Using this method, the size of DNA fragments was determined because the rate of movement of a molecule in gel electrophoresis is linearly proportional to the number of nucleotide base pairs. Then, to observe very small amounts of DNA, they were stained with safe stain, which glowed with the property of fluorescence when exposed to ultraviolet light. The results obtained from PCR arms were analyzed using SPSS software, and analytical analysis was performed on the data. Data were analyzed using Chi-square statistical test. In this study, 200 women with infertility and 200 pregnant women controlled, who were the same in terms of age, sex, and geographical location, were studied. Subjects were compared in terms of genotypes of both interleukin 22 gene polymorphism. The result of examining the frequency of each of these factors showed that ovarian laziness had the highest percentage, and people with thyroid hormone problems had the lowest frequency of causes of infertility in our study population. Using PCR technique, interleukin 22 gene fragment was amplified in two loci: in gel electrophoresis, a 361 BP band in polymorphism polymorphism first, and a 276 BP band in polymorphism second were the product of PCR. The 106 9 BP bonds in all samples were the product of the control gene primer. In the first variant, 1361 BP band represents the homozygous JG or double A genotype, and 361 BP bands represent the heterozygous AG genotype. In the second variant, 276 BP band represents the homozygous genotype of double G and double A, and 276 BP bands represent the heterozygous AG genotype. In the first place, in the patient group, P equal 0.001 x2 equal 7.3, and in the control group P equal 0.001 x2 equal 11.2, which indicates the relationship it was significant and the population was not in balance, and the results showed that there was a significant difference in the genotype between the patient and the control group. But in the case of adults, P equal 0.939, which was not significantly different, and there was no relationship. In the second place, in the patient group, x2 equal 7.4, P equal 0.005, and in the control group, x2 equal 8.7, and P equal 0.005, which showed that there was no significant relationship between population and balance, and there was a significant relationship in genotype between patient and control groups. In the case of owls, with P equal 0.433, P greater and 0.05, no significant difference. To investigate the effect of owls in dominant, recessive or heterozygous conditions on infertility in polymorphism, first statistically tests were performed, and according to the all three genotypes had a significant relationship with infertility, with P less than 0.05. In a second polymorphism, double A and AG genotype with P less than 0.05 were significantly associated with infertility, and double G genotype was not significantly associated with P greater and 0.05. Logistic regression test was performed in both patient and control groups. Data analysis showed that the overall result of combining the genotypes of these two loci with P equal 0.001 in both patient and control groups was significantly associated with infertility. Examination of gene combination compared to other compounds in two polymorphism showed that out of 9 genotypic compounds, 2 ratios of AG-AA and AG compared to other compounds in both above positions with P equal 0.026 and P equal 0.0, P less than 0.05 was significantly associated with infertility, and other compounds were unrelated. According all clinical factors with both variant of interleukin 22 gene had p equal 0.0, which were significantly associated with infertility. That is, these factors increase the likelihood of infertility with these two polymorphisms. In this study, for the first time, the relationship between two genetic variants of interleukin 22 molecule at first and at two variant loci with female infertility was investigated. Their genetic information of the above gene and this polymorphism can be used as an indicator for screening and prevention of people at risk of infertility, as well as in designing new treatment guideline and predicting the recovery of patients. According to the statistical result in the position first bond pattern obtained from PCR DNA extracted from the blood of 200 people in the control group. 177 people had AG genotype, 14 people had JG genotype, and 8 people had AA genotype. And in the group of 200 patients, the frequency of genotypes was order 151, 29, 18, and G-score test was performed to investigate the relationship between genotypic frequency in this locus and infertility disease, and the result of the analysis showed a significant relationship, p equal 0.0 and p less than 0.05, and statistical test for allelic frequency was performed in two groups with 15.8% in the study population had allele frequency G, and 48.2% had allele frequency A, and in the patient group 15.2% had allele frequency G, and 48% had allele frequency A. Given that p equal 0.939 and p greater and 0.05, G-score test didn't show a significant relationship between allelic frequency at this site and infertility. Therefore, a possible relationship between the genotypic frequency of this site and infertility was confirmed. In second polymorphism, out of 200 controls, there were 175 AG, 15 AA, 10 GG, respectively, and in the group of 200 patients, 148 AG, 13 AA, 90 GG. G-score test was performed for the relationship between genotypic frequency of this locus and infertility, which was p equal 0.0 and less than 0.05. Regarding allelic frequency in the control group, G 48.2% and 51.8% A, and in the patient group 45.5% G and 15.4% A, and P equal 0.433, which was P greater than 0.05. The above results somehow confirmed the relationship between genotypic frequency of this site and disease, but relative frequency was not significantly related to infertility in this site. In human, interleukin-22 has a dual role in tissue repair, host defense, mucosal protection, and autoimmune disease, and its low level causes inflammatory disease and is similar to pro-inflammatory cytokines such as interleukin-10. However, these two interleukin-22 gene polymorphisms in the 5-UTR region can trigger the activity of amplifying mRNA such as 192, 194, and 122 in this region, causing splicing and intron depletion. As a result, the axons are joined together, and the target mRNA is isolated possibility. Finally, the expression of the interleukin-22 gene increases and its serum level decreases in the body. And the interleukin-22 gene in this case acts as a pro-inflammatory agent according to the studies performed in this study in two groups of patients and controls. It was observed that individuals with AG genotypes, if they have both polymorphism in 5-UTR, the interleukin-22 gene increases their risk of infertility. In first polymorphism, the amount is P equal 0.001, and in polymorphism second was P equal 0.005, which considering that in the fourth position. The correlation intensity for AG and GG genotypes were 3.4 and 1.4 respectively, and in the second position for the same genotypes the correlation intensity was 2.6 and 1.1 respectively. Therefore, it can be concluded that these variants were associated with infertility, and statistically this relationship was meaningful. In this study, the confidence in interval of the fourth place for AG and GG genotypes were CI equal 1.4 till 8.23 and CI equal 0.4 till 4.17 respectively, and in the second place and the same genotypes were CI equal 1.138 4.89 and CI equal 0.4 till 3.1 respectively. Therefore, it can be concluded that these two polymorphisms were significantly associated with infertility in all three genotypes with P less than 0.05, and according to our greater and 1. These two polymorphisms were associated with infertility. The role of risk factors and clinical factors such as ovarian laziness, uterine problems, endometriosis, in disease 2 situation, interleukin 22 genes was effective in increasing the susceptibility to disease, and these clinical factors were significantly associated with infertility. Thanks you to all the professors and colleagues for allowing me to be at your service. Good luck.